Recording, maybe. Yeah, because we used to do this on Blackboard Collaborate and that would automatically upload the recording. But because we are doing Zoom, it won't be done automatically. So I think it's at the discretion of the prof. And the rest of the tutorials will be online too. Yes, it will be online. All right, it is 10:10. So we will get started. How is everyone today? Awesome. I'm good. Thanks for asking. Okay, so let's start off with some quick intros. Before anything, this is the agenda for this tutorial today. So we will do intros and then we'll go over like a quick schedule and then how to read a paper. And then we're going to quickly go over the paper that you have to do the reflective piece on. And then we'll discuss some of the expectations that I will be looking for so you can get that A+ grade on your assignment. Okay, so just a quick fun fact about me. I'm Sandy. I'm going to be your TA for this semester. I actually did my undergrad here at U of T, also doing a major in health and disease. My other major was cell and molecular biology. God forbid. That was painful, but you know, we make it through. And I currently study like molecular markers of tumor hypoxia, so cancer research. And my favorite snack, honestly, I have so many, but I would say gelato, you know. And I have a small Maltese at home. And if you have any questions about the course content, life, grad school, you know, any career related questions, you can also reach out via email. I respond pretty quickly unless I'm dying in the lab. So because I also want to get to know you all, I would love for you to kind of write out what your majors are, what your favorite snack is. If you have a pet, and if you don't have a pet, your favorite leisure activity to do in the chat. And I just want to also preface by saying your participation in the class matters, and this will also be part of your tutorial grade / reflective assignment grade. So make sure you participate. And this is a great way to get your participation grade for this tutorial. And thank you to those who have already sent in their fun facts. Yes, Shreya. Amazing. Wow. I feel like some of these programs I've never even heard of before. Like, is it new? Oh, I love Triscuits. Lay's ketchup chips. Yes. That is my guilty pleasure. Absolutely. Wow. German minor. That's intense. Oh my God, these are so fun. I love it. I have a dog. I also, oh my gosh, should I drop my dog's Instagram so y'all can follow? Okay, so it's at MOCHI dot EMOCHI. Wow, all my lab members are judging me so hard, but it's okay. uh, but like totally go follow her. Okay, now I actually get into the tutorial. Okay, and if you also have a pet that has an Instagram, drop it. We will all follow each other. I also named my cat after food. Her name is Cream Puff. Honestly, the best. Thank you. Thank you. Thank you. Okay, I love it. Okay, now that we're all hyped up, let's like actually talk about science. Okay, so I have some questions for you on how to read a paper. How many of you have actually read scientific literature before this class? The answer should be yes, because of Bio 130. Yes, that is the correct answer. You guys probably read about plants a lot. I don't know, some microorganism from Bio 130. Am I right? And Bio 120. Yep. Okay. Can you tell me that some of the main steps that need to be taken when you're reading a paper? Like, what's the first thing that you should technically read when you're reading a paper? Okay, after the title. Yes. So the abstract is a myth. You should always go to the last paragraph of the intro because that will give you the best overview of the entire paper, including the purpose. So in that last paragraph of the intro, you will actually get a very very solid outline and what kind of studies that they did and the summary of the main results in that last paragraph. So if you're looking for a paper that answers certain questions that you're wondering about, that is a great place to start when you are screening a lot of papers that you want to pick through. And then after you have decided that, okay, maybe you should continue reading this because it's somewhat interesting and you want to learn more about it, then you read the intro to get, the background information and to understand the gap in knowledge. And then you would go to results. So you can just skim over the methods when you are reading the paper, mostly because you're not really focused on the nitty-gritty details of the method section, okay? Because frankly, like at this point in your career, you're not going to be, the point of reading the methods is to understand how it was done, to recapitulate the results yourself. But for, in a course, it's more important for you to understand exactly what was done, like why some of these methods were used to answer these questions. So, for example, when an author uses a Western blot, the whole point of a Western blot is to measure protein concentration, right? So that's kind of the main message that you want to really look for, rather than what concentration of all these different like antibodies and like different chemicals that are used. Okay. And then after you've read through these amazing scientific discoveries, you would want to look at the discussions and the conclusion section. Any questions on the blurb that I just talked about? No questions? I think it's pretty straightforward. No questions? All good. Okay, thank you, Jake. And of course, in in every literature, there's always going to be a hypothesis that's going to be addressed. Can anyone tell me what a hypothesis is? How can you define a hypothesis? Okay, those are pretty good guesses. Yeah, so obviously every literature will have a research question that has to be addressed, right? But a hypothesis is something that can be testable. So yes, it's a testable statement. So always remember that when you are reading the paper, and mind you that in every literature, it's not going to be specifically identified as our hypothesis is, or this paper aims to hypothesize X. Sometimes it will be worded differently. So even if it doesn't say specifically what the hypothesis that they're testing, you should be able to pick it out by the way that the paper is written. Okay. All right. So before we get actually into some of the questions that you should answer for the assignment I believe it's due this week, Sunday, right? Am I wrong? Yeah, Sunday. Okay. I just wanted to give you an outline of the expectations of what I'm looking for when I'm marking these assignments. So I know that there are like 5 major questions that are on the assignment sheet, right? I think 5 or 6. Please answer those questions. You do not have to write it in full like essay format. If you just want to write out the question and then write your answer right below it, that's good enough for me. I just want you to really understand what the question is asking and provide me with the answers that you think is correct. And make sure when you are writing these assignments, there are no jargon / flowery language. Scientific writing is not about how beautifully you form your answers. It's about getting to the point. The question should be available on Quarkus. And also tell me what you think. Do not really copy and paste exactly what's already written on the paper. Tell me what your discussion points are and how you can make this research better. And if you can answer these questions with critical thinking and critical analysis portion, it doesn't really have to be a full page. If there's a word limit, only our answers will count for the word count. Yes, it is absolutely correct. So if your entire answer, like only the answers copy and pasted in a full page, that's good enough for me. And then the format is the following. Use Arial, font size 11, margin 2.54, single space, citation style would be in text. Any questions? And thank you, Sophia, for linking the reflection sheet. Just to confirm, it's due Sunday at midnight. Yes, it is due Sunday at midnight. Yeah, Tala?
I just wanted to ask, so when you said, like, when you put in the bullet points, tell me what you think and how can you make this research better? I was just wondering, how can you make this research better, like an additional response to the 5 questions, or where would this lie in the 5 questions?
So I would say it should be number 3. What kind of approaches are being taken to address the questions posed? And so you want to answer that, and then to make it even better, you would tell me what other experiments can be done to address the questions that are being raised by the author. Because I really want you to look through other literature to support what they are doing. Or rather, if you think they're doing something wrong, tell me and propose something for me. Does that make sense? Yes?
Yes, thank you.
Okay, no worries. All right, so when I go through how the mark breakdown is going to work, it'll make a lot more sense. So I personally do not believe in failing anyone, for the purpose of everyone's sanity, and I just don't think it is productive for your learning. But if you are getting the 6 to 7 range, that means that you have a lot of work to do. Mostly because you have incomplete and incohesive sentences, questions were not answered properly, there was no critical analysis, and no references. Yes, Carmen. And yes, you do need a works cited every time that you have an assignment to do. And 7 to 8 is incomplete/incohesive sentences. Questions were attempted but not answered fully, and there was no critical analysis that was performed. And then 8.5, that's when you do the bare minimum, which means that questions were answered fully and the critical analysis was attempted but was done poorly. So, for example, an example of that is when you just cite another paper and you think that that's going to be a critical analysis. A lot of the students do that. You don't want to do that. So, for example, in this paper, in a paper that you're reading, they found that protein X was regulated and activated some molecular pathway that's involved in stem cell formation, right? And then your attempted critical analysis would be like, oh, and another author performed the same study and showed this. No, that is not a critical analysis. That's more of an attempt. You want to really talk about what the other author did to support your current author's finding. And then 9 is questions were answered fully. Critical analysis was present with a single source. And then 10 out of 10 is, of course, with multiple sources. I'm going to say that the first reflection piece, I will mark easier than the other ones because I know that this is your first rodeo. But second reflective piece onwards, it will be marked a bit more difficult. Is that fair? Perfect. Yes. Because I want you to have a chance to, realize where you're missing and all those things. So your first mark will not be fully reflective of this because I will mark easier. But I will still tell you where you were missing marks and what you would have gotten if I marked normally. All right. Okay. There are no sample reflective pieces. That's why I'm giving you the first time to, give it a go. Sounds good. Okay. Now we're going to answer some guide questions, but I'm going to stop sharing my slides. So my first guided question is, Can anyone tell me the major result of the study for the paper that you were assigned for this week? Well, I guess better questions to start off is, did everyone read the paper? Okay, so these questions should be very easy. I'm going to copy and paste the guided questions in the chat, and then you guys can start answering. Oh, I don't know why that was copy and pasted like that. There you go. That's better. Okay, Nicholas said patients with influenza A release airborne particles containing. Okay. Perfect. What type of specimen was collected? Perfect. And then, which assays did the authors use to test the research question? There are 3 different assays that were done. Viral plaque assay, viral replication assay, and we're missing one more. We're missing one more. No. Yes, qPCR. Yes. Yes, 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 yes. Okay. And how can we limit reducing the spread of airborne viruses? This should be very easy because, you know, COVID. Yep. Slip PP, isolation, ventilation, yes. Filtration, respirators, patient cohorting, and getting shots, yes. And can you tell me some pros and cons of using informal sources to track transmission based on the paper? What are some of the pros? Less expensive, faster, yep. And cons, right? Might be misleading, inaccurate. And also, it is cost effective. Beautiful. All right. Well, I hope these guided questions will help you answer some of the questions that are on the reflective piece. But I think that is it for today. If you have any further questions, you can stay and ask. But if not, it was so nice to meet you all, well, virtually. And I will see you next week. Thanks, everyone. Bye.
Hi, I just have a fast question.
Yep, ask away.
Yeah, it's not about the paper, but I wanted to ask about research in undergrad. If
Sure.
Yeah, I was just wondering where do you suggest starting, like for research and all?
Email me and I'll send you some links.
Okay, sounds good. Thank you.
Okay, no worries. Sharia?
Hi, Sandy.
Hi.
I had a question. If you could go back to the slide or like type your email in the chat, I forgot to note it down.
Yep. I am doing that right now.
Thank you. And also, so I don't know about the like population of the class, but I'm third year. So if I have to like, if there's something that I know from just studying the material in class, is that material I still have to cite?
No.
Okay. Okay. So like if I'm talking about the design of a study or like limitations and cons or like types of studies that I think would fit better, those are not types of things that I need to cite, right?
Nope.
Okay. Okay, perfect. Thank you.
Thanks.